Okay, so I guess most of the people have changed now, so I would like to start the session now. We're just in time. We were dealing with the question, what are the hurdles still getting cancer therapy or getting nanomedicine into cancer therapy and getting things from the production side. For us, this is here the Max Planck Institute for Polymer Research into the clinic. And that is, well, I chose three things which I wanted to talk about shortly. And this is targeting in its hurdles, and we have seen this 0.7% of targeting or any nanomedicine that's getting into the target side, which is very disappointing. Then is there an in vitro and in vivo correlation? Do we always think about it? How can we think more about that? And then the question is, can we indirectly target a tumor? And I think we've seen just before and in some other settings as well that we can also do that, but we will have a lot of talks also afterwards where we directly target a tumor still. So why do we do that? Well, when I came into that field, of course, I came as a medical doctor, somebody who has been treating patients with leukemia, lymphoma, and solid tumors. I was thinking, well, we need to distribute our pharmaceutical agents differently, and nanomedicine would be a great opportunity to do that. We could change the pharmacokinetic of that, because when we basically take the pharmaceutical agent, the active drug, then we flood the whole body. And that's when you work in clinic, you know what that means. It means that you have a low amount of your target, of your drug in the target region, and then you have the rest makes your side effects, hair loss and things like that. But not only hair loss, sometimes you need to decrease the amount of dose you inject, and if you decrease just a little bit, then you lose even more effectiveness. And vice versa. If you can increase that, maybe you get more. And there are, and we have seen it before, RNA species like siRNA or the messenger RNA. If you use them, if you use small peptides, they may be extremely unstable. They maybe need something to package that with. And the promise was, well, we can target it all to one organ, to one specific, 100%, so to speak. So that's always how we look at it, right? And for some systems, it seems to work. We've just seen that we can target the lung very effectively, for example, or the spleen. But for most particles, it will not be like that, I guess. So it has a high capacity. We can protect the cargo. I think that's really something that nanomedicine has really approved, that concept. We can go into cells with molecular drugs that would not enter cells by themselves, like messenger RNA, like siRNA. And we can have it as a platform technology. Not always, but sometimes you can use it for more than one. And we can combine things. This is something Twan Lammers is talking a lot about. And we can have also something like a slow release. But let me talk a little bit about how can we target that better. I think that is a question that we still look into and that I was thinking would be much easier, but has turned out to be much harder. So targeting and its hurdles. Why? Well, basically in 2013, Dawson and the first author is Salvati, who is also here, and they have pointed towards why that is, because we have proteins on it. So is there still targeting if we have all that proteins on the nanoparticle? Because then it's covering everything up. Does that do unspecific uptake? And it does. Would that be also do that on a macroscopic scale? And so, like shown before, and like what you read in literature, you would say, okay, if there's unspecific protein absorption and you get unspecific uptake in the macrophages and things like that, why don't we just decorate them with stealth polymers? And then we don't have like polyethylene glycol, and then we don't have any proteins anymore. We don't have any cell uptake anymore. And we also, of course, that's something we did as well. So we used here polyethylene glycol. So you see that two different types, 44 and 100 units of that EO, and then also a polyethylene ethyl phosphate as a experimental particle. And what we did is we incubated once with a cell culture where there was plasma in it, and once we did just remove the plasma. Why did we do that? Because we said, well, if it should be even get better, right? Or it should be at least have the same results if we don't have proteins on it, and if the lack of protein is giving us this non-uptake effect. But of course, it's not like that. And when you look a little bit closer, you could also reverse that and say, well, The particles that have no uptake or less uptake, they only have that because of proteins. And when you then look what proteins are there, you find one protein of all of them, and it's very prominent. And to make a long story short, we did a lot of 2D gel electrophoresis and things like that. But we also looked what it is, and it's actually an apolipoprotein J, also called clustrin. And the other apolipoprotein proteins are all there. Also there, apolipoprotein A1, which is an HDL protein. So our paradigm from now on was we have a reduced but selective absorption, and actually this protein which is absorbed is then hindering the uptake. And the benefit of that is we can then screen other materials for this stealth effect and look just for clustrin, ApoA1, and the other apolipoproteins if they're there or not. And if we do that, we can have a low uptake. So we see the first two bars here, low uptake for the non-targeted ones. But if you have here we do a target with monos and tremonos into a dendritic cell line, and even if you use that 100% serum inside your cell culture, you can still do that. The cells don't so like that very long, only for 1 up to 2 hours you can do that. There's another target, or there's another hurdle to it, and I'm not going to speak about it because a lot of people will speak about it afterwards. That's the EPR effect, but it's not going too deep into this, and this has been something that Juan Lammers has been publishing a lot on, and I think this is something we will hear in the next talks about. So is there an vitro in vivo correlation? Well, when we were working with this protein corona and targeting, we had been starting working with serum, of course, for a long time. But we said, well, serum does not contain coagulation factors, not fibrinogen, of course not the cells, but that's also true for the other. If you put an anticoagulant in that, you also get rid of the leukocytes, thrombocytes, the erythrocytes, but you have all the other things in there. But, of course, you add something. And you add something like EDTA, citrate, so you complex calcium, or you have heparin in it. So you add something. Does this do anything? Can we then get different results? Does that give you, do we always get the same results? And here you see, well, we have this HeLa cell, or you could take any other, many other cells, your favorite tumor cell line, and then you have fetal bovine serum, human heparin citrate, and human heparin plasma, and human citrate plasma. And you see, well, there's a good anti-uptake effect with that. But if you use a macrophage cell line or primary monocytes, then you see that with the human heparin plasma, the HHP, that there is, again, an uptake. So is heparin promoting that uptake? Is it by itself doing that? With the HeLa cells, you can see if you use FBS plus heparin, it gives us the same effect. Actually, there's less uptake, so there's an anti-uptake effect of heparin itself. And for the macrophages, there's, as we thought, there's actually an enhanced uptake. So the question is really what to use. And we have seen also other examples, and last year I was telling here that we may use citrate plasma, but what we do these days is that we use different kinds of plasma, like heparin plasma, citrate plasma, and also serum still, and compare that and correlate the results, and correlate that to the protein corona we find, and that we should correlate that to in vivo results we are acquiring. So can we do an indirect targeting? Because a lot of people will speak a lot afterwards on a direct target, but we have just seen that we can, by Mr. Haas, that we can very nicely get into the cells of the blood and also into the endothelial cells, but I'm going into the immune system now. Well, when we look at that promise that we directly target the tumor, that we may still do, but there will be other people talking about what we can do then afterwards. And what we think now, where also Mr. Haas is involved, is that we should target the immune system. So it's basically the dendritic cell which readily takes up nanoparticles and can then instruct the other cells like the cytotoxic T cell or the NK cell in order to get to the tumor and destroy the tumor. So it does not need to be really there. It doesn't really need to be there, and that's also something that's now funded by a collaborative research program in Mainz in order to do that. Cells are in the blood, the spleen, or in the lymph nodes, so they are more easily targeted probably than the primary tumor. You can have an active targeting with antibody or ligands, and there may be even cells where you don't need it, like for dendritic cells, which just take up things by size or by other physical-chemical properties. And you have, of course, also the multiplication effect of the killer cells. And immune cells also find metastases you're not able to reach by nanoparticles, because you need to get through a barrier, and some CTL, some cytotoxic T cells will just do that for you. And, of course, you could combine that very easily with other popular treatment options like checkpoint inhibitors these days. And this you have seen before, actually. Mr. Haas is presenting some data from that Nature article. So that is really something that I think everybody should read about. So how do we bridge that gap? Well, we have been learning a lot about nanocarrier production and development. We know a lot what we need in medicine, but we don't understand necessarily what's in between. We don't understand what's going on the cellular level, on a subcellular, intercellular level, how things are processed, how things are getting out of the endosomes, for example, and of course, what also goes on the organism level, and still then also translating that from mice to men is a big hurdle, or is a challenge for many things, but it's much closer than just having a concept and evaluating that in cells, definitely. So understanding the complex system, even more complex surrounding like an organism, I think we need much more in ex vivo and in vivo correlations, and I think we've seen some of these correlations these days here, and also going from animals to humans, and we've seen that a lot of the data or some of the data is generated is also now in non-human primates. So with that, I would like to thank you for your attention, and I think I can take one or two questions. Thank you very much. Any question?
Thank you for a very nice talk. I was triggered by the corona, actually, that you show that it's forming sort of like a selective, maybe unspecific. But do you think that the size and the charge of the nanoparticle and the material will make the corona, which will be specific or which will be forming or acting as a ligand for certain receptors expressed on macrophages?
That's something people like to do, is that I say I make a physical chemical surface, and then I try to attract specific or wanted proteins that then target my target cell. It will form the same same protein corona whenever you make exactly the same surface, and I think that's the hard thing, really, to have exactly the same physical-chemical surface, to have the same amount of functional groups on the surface, and that is something that needs to be related very well to from batch to batch and very well evaluated. And sometimes we see changes in the protein corona and go back to the chemist and say, well, there must be something wrong. Can you measure that again? And when they look very close, they say, well, there's a little bit of difference, but do you really see that? And yeah, we do.
We have done some experiments, and we have published a paper in Nanomedicine recently where we looked for M1, M2 uptake of silica nanoparticles with and without plasma. And we see that the proteins are adsorbed on the surface of larger nanoparticles. We started with 50 to 100, 50, 100, 500, and 1,000. And in 1,000 or 500, they were like corona, which was taken up, making the particles taken up by M2 types macrophages, and smaller one in M1. Does it happen also for other cells type, like that kind of thing? I was wondering, like in dendritic cells or other cells, T cells, probably.
Yeah, I think I've more experience with the dendritic cells, and they go even higher. You can go give them particles, feed them up to particles with 250 or 300 nanometer in diameter, which maybe not taken up the other cell types, and they may definitely go through. So size is definitely still a big question, and also the amount of, or the composition of your protein corona will change in that phase. Okay, I think then we need to go on.